Hey everyone, this is Dr. Randy Papke, and today we're going to talk about Chapter 6, DNA and Gene Expression. Just so you know, I've got a bit of a cold working, and I might clear my voice or cough a little bit, and we're just going to play it by ear and see how it goes. So, Chapter 6 is all about DNA and one of the main jobs of DNA, which is directing gene expression. Basically, gene expression is just the way the cells make proteins. So we'll learn about a couple of steps involved in making proteins: transcription and translation. And then we're going to talk about how mutations actually cause the DNA to change and what causes those mutations to happen in the first place. And then we'll talk about like some of the effects of those mutations. So, you know, a lot of people want to know about why they need to learn about DNA. And it turns out these days DNA is everywhere. It's like so important to know, especially when it comes to criminal justice. So this is a gentleman here. His name is Julius Ruffin, and back in 1981, he was working at this school. He was a maintenance worker, and he was accused of raping somebody. And it turns out that the person who accused them honestly thought this was the person who raped her, but it was basically eyewitness testimony. We've already talked about the downside of eyewitness testimony and how that can be so non-scientific, just not useful, not reliable. So even though, excuse me, his girlfriend testified that he was with her at the time, this eyewitness testimony convicted him. But you know, a long time passed, and eventually people started doing DNA testing on these old rape kits, and it turned out that that he was freed because there was literally no DNA evidence that he was the one who did it. 344 people have been released from jail who had been unjustly imprisoned. So that's a pretty big deal. Spending an average of like 13 years in jail in prison for something that they did not commit. So the more we understand DNA, the more we understand how it works, and the more we understand its use in the criminal justice system, and how it's used in later chapters, how it's used in agriculture and so forth, the better off we are. So, besides criminal justice, we have DNA in the news. So we hear about how you know, are your genes responsible for or what you do, or how you behave, those kinds of things. So it's important to know about DNA. The structure of DNA was figured out by Watson and Crick. I'm really unhappy that your book also doesn't spend a little bit of time talking about Rosalind Franklin and her important role, but we've known since the 1900s that DNA was kind of the molecule of inheritance. It wasn't until the 1950s that we figured out how it worked. And lots of scientists were involved in this. And we're not going to go into the details of any of them, but it took a concerted effort of many, many, many scientists to figure out the structure of DNA. And I'm going to talk to you about that all basically on the next two slides. And there's a lot of information on the slide, so I'm going to be using my mouse to point things out and just kind of going through this diagram and having some reinforcement on the next slide so that you can make sure you've learned everything. And there are lots of terms on this slide, so make sure if you're taking notes along with me that you're writing down the terms. Okay, the first thing is that DNA is actually a three-letter acronym for deoxyribonucleic acid, right there. Oops, sorry about that. Deoxyribonucleic acid, and deoxyribonucleic acid is a very, very large molecule. It's one of the largest molecules that exists. So it is actually made up of a bunch of smaller molecules called nucleotides. So there's your term right there, nucleotide. And nucleotides are made up of, themselves made up of, the smaller parts. So what are the smaller parts of the nucleotide? There is the phosphate group. right here, phosphate. And phosphate always looks the same. It's phosphorus and these oxygen atoms here. And then there's the sugar group, which looks like this. It looks like kind of like a pentagon. And the sugar in DNA is always known as deoxyribose, which is how deoxyribonucleic acid gets its name. It's actually named after the sugar. Together, phosphorus and the sugar deoxyribose make up something called the sugar phosphate backbone. And you can see that term right there, the sugar phosphate backbone. The thing about the backbone, if we look at the big molecule, it's like this ribbon right here. It's the ribbon. And that ribbon is important because it holds everything in place. But what it's holding in place are these nitrogen-containing bases. You might also hear me say nitrogenous bases, or sometimes I'll just call them bases. So these nitrogenous bases or nitrogen-containing bases, you can see they have these letter N's in there. There are only 4 of them. There's only one type of phosphate. There's only one kind of sugar in DNA, but there are actually 4 kinds of bases, and adenine is the name of one of them. We give it the letter A, and you can see the letter A right here to indicate that it's adenine. So, so this is the nucleotide right here. Those 3 ingredients make up a single nucleotide, one nucleotide. And if you string a whole bunch of these nucleotides together, you're starting to get DNA. So let's take a look at what the bases look like in their variety, because there are 4 different kinds. And there's adenine right there, and you can kind of see it's a simplified version of what we saw up there. And adenine, always, 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 base pairs, that means lines up with or like connected with these little dotted lines. Those are hydrogen bonds. And there's your term right there, hydrogen bonds. These little hydrogen bonds hold them together with thymine. So adenine always base pairs with thymine, and thymine always base pairs with adenine. And you can see that over here on the molecule. A goes with T. Every single time you see an A, you'll see a T over there. And in the end, what that means is if you know how many adenines you have in a molecule, you actually know how many thymines you have in the molecule. Guanine, G, always base pairs with cytosine C, and actually has 3 sets of hydrogen bonds right there. So guanine and cytosine always base pairing. There they are, always base pairing. Doesn't matter which side it's on, guanine, cytosine, base pair. So we call DNA a double helix because there's 2 backbones. There's one backbone right there, and then there's this other backbone on the other side right there. And they don't ever really touch. They're just held together by these hydrogen bonds that are connecting the base pairs together. So that's your basics of DNA. And I just want to summarize really quickly, like DNA is a nucleic acid. It stores information, and it has these individual subunits called nucleotides, which are made up themselves of sugar, phosphate group, and a nitrogen-containing base. We got your double helix. That I didn't say earlier, but that kind of looks like a twisted ladder, and the sugar and phosphate are on the outside, and the base pairs are like the rungs of the ladder, the inside. Another term that I didn't use earlier is the term complements. And that's not saying, hey, you look nice today. It's complements is more like a mathematical term. You know, 2 angles are complementary if they add up to 180 degrees. So here we see A and T, they kind of complement each other. They always kind of make this line together. So complements. One of DNA's like really fantastic features is that it has all the instructions for building cells, and almost every single organism on Earth is going to have this DNA. There's some only weird little exceptions. For example, like red blood cells don't have DNA in them, but DNA is also the universal language. Everything that has DNA, and again, it's almost every single cell on the planet, has A, T, C, and G. It's a universal code. You have the same A's, T's, C's, and G's in a different order that onions have, that fruit flies have, that amoebas have, that salamanders have. Everything has this kind of information. And so, so we have a lot in common with all life on this earth. If we're looking at DNA and all the DNA that's found in a single organism, we use the term genome. So when I say all the information in a single organism, I really mean all the DNA in a single cell of that organism. Because every single cell in your body, again with the exception of the red blood cell, every single cell in your body has all this DNA in it, and we call all that DNA in that cell, we call that the genome. Okay? So all the DNA in that nucleus, that's the genome. Now, the DNA doesn't all come in like one molecule. It's actually divided up into many little molecules, or well, smaller molecules, I guess. And those smaller molecules are called chromosomes. Now, we've already talked a little bit, I think, about how confusing the term chromosome is, and we're going to continue to kind of deal with a little bit of that ambiguity. But for now, chromosome is one or more unique pieces of DNA. In the case of prokaryotes, that DNA is going to be circular. In the case of eukaryotes, that DNA is linear, meaning it has an end and an end that are not connected to each other. The chromosome literally has hundreds of millions of base pairs going on there. If we take a close look at just one little section of that chromosome, we might find something called a gene. And genes are really important in this chapter because genes are the sequence, is a really specific sequence of DNA that codes for proteins. You cannot define gene without saying that it codes for a protein. It's not just any specific sequence of DNA. It's the sequence of DNA that codes for a protein. The term locus simply means it's a position on the chromosome, is a position of that gene on the chromosome. So we could say that this is the locus of the gene. Maybe there's a locus over here and a locus over there, if that makes sense, for different chromosomes, for different genes. We humans have 46 chromosomes total. But we got half of that information from our mom and half of that information from our dad. And so we use this, it's a little strange, the way we use this term. It'll become clear a little bit later when we start talking about cell division. But for now, we say that humans have 23 unique chromosomes, 2 copies of each. So we can also say humans have 23 pairs of chromosomes, one from mom and one from dad. 23 pairs, 23 times 2, that makes 46. And hopefully that makes sense. You've heard gene used before. It's kind of mysterious, but we'll come back to that and really get a good handle on what that means. So along with this idea of genes, and remember genes are at a specific locus, right? And remember that you get some information from your dad and some information from your mom, and this is, pretty true, doesn't matter what species you're talking about as long as there's a dad and a mom present. But remember that your dad and your mom don't look exactly alike, and they can give you different versions of a gene. And the word that we use to describe different versions of the gene is called alleles. That word is pronounced alleles. So different versions of a gene. If we look at this gene at this locus, this gene is the gene for flower color. And in these plants, there's 3 different versions. You can have the orange version, and you get the orange flower. You can have the yellow version, and you get the yellow flower. And you can have the purple version, and you can get the purple flower. All I'm trying to say here is that those different versions are called alleles, and that's such an important term. Actually, the term alleles, even though it's a little challenging, it's a lot like, well, it just comes up in so many different chapters that you really have to understand it. So different versions are alleles, different versions of genes are called alleles, and a trait is what that characteristic looks like or appears like. So for example, this one looks orange, or you could say somebody with the allele for cystic fibrosis has the trait of cystic fibrosis, if that makes sense. It doesn't have to be appearance, but it definitely can be appearance. So what do I want you to take home from all of this? DNA is absolutely the universal language. It builds structures, it builds organisms, it builds proteins. All of that's called the genome. In a single cell, it's called the genome. Prokaryotes have circular DNA, eukaryotes have many chromosomes, and that those are, in fact, linear. And then, of course, finally, we get into what a gene does. And we're going to get into a lot of details about what a gene does. But for now, let me just give you a heads up that we're making proteins. And one special name for a protein is called a polypeptide. That just means lots of amino acids. Or you could also be making RNA molecules, and we're going to talk about how that's pretty essential in the making of the protein as we go along. So, and we're going to spend a huge amount of time talking about what happens with genes. But it turns out, and I think this is really, really cool, it turns out that not all DNA is made up of genes. In other words, not all DNA is made up of instructions that directly make proteins. So here's a cool little diagram. If you look at the genome size of various organisms, fruit flies, they have 18, and this is a little confusing because it's in millions of base pairs. So this means they have 18, I think that's supposed to be 180. I think this is a little bit of a typo. 180 million base pairs. Humans have 3,400 million base pairs. Another way to say that is they have 3.4 billion base pairs. Onions have 18 billion, salamanders have 34 billion, and amoebas have over 600 billion base pairs. The size of this line, though, it's just not in proportion. We had to have this little break here. So amoebas have way more DNA than fruit flies or even humans. So that's the first thing. If you then compare how much of that genome is actually made to make proteins, in other words, how much of it is actually genes. In humans, only 2% of your genome is genes. The rest is something for now we're going to call junk DNA, and we're going to, in the future, after this slide, we're going to call it non-coding DNA. It was known as junk DNA because we didn't know what it did for a really long time. So 98% of the human genome is junk or non-coding DNA. In fruit flies, it's 19%. We can kind of go all the way up to E. coli, which is a type of bacteria. I hope you know that. And this bacteria, 90% of its genome is actually specifically made for making proteins. So this is a little weird. And for a long time, scientists did not know that what was going on here. So let's kind of just leave this behind for a second. We're going to find out what some of this junk or non-coding DNA does, but let's get a little closer view on what that actually means if you're looking at genes. So coding versus non-coding. Again, we're going to leave the term junk behind for now. That was the original name for it. So we do know that bacteria, viruses, that 90% of their DNA is coding, but eukaryotes, much, much less. So let's take a look at what that looks like. If this is a gene, if this is gene number 1 and this is gene number 2, what we have here is the space between the genes. And the space between the genes, we call that intergene sequence. That term isn't right here. We'll talk about it more in class, but that's your intergene sequence. And some of that intergene sequence, you're going to find things like promoters, other things like that that we'll get to really soon. But interestingly, within a gene, and here's the beginning of the gene and here's the end of the gene, within a gene, there are also regions that do not code for proteins. And I know this sounds weird. And so we're going to have to deal with these regions. Remember that these regions were called intergene sequences. And these regions have a name as well, and that name is intron, which I do have here for you. Introns are non-coding regions within a gene. And so there's an intron right there, and there's 2 introns right there. Again, we will talk about introns, and we'll talk about how we deal with introns as we move forward. So So one of the things, just as a heads up, that we know about these intergene sequences is that they're really important in figuring out, for cells, to figure out what kinds of proteins they're going to make. So, for example, your body has the DNA that codes for making your eyeballs, right? Your eyes. It also has the DNA that codes for making your heart. Why don't you make eyeballs in your heart? The DNA is the same. Like, you have the same information throughout every cell in your body. And the reason for that is these intergene sequences. So we'll talk again more about that as we go along. So let's take a quick look at a few terms that are going to be important throughout much of the rest of the semester. And that's genotype and phenotype. So a genotype really talks about the DNA. It's really looking at the genes, specifically which versions of the genes you have, in other words, which alleles you have. So genotype is which alleles, which combination of alleles you have. Your genotype affects your phenotype, and the phenotype is this physical manifestation. In other words, it's kind of like your appearance. But I like the way phenotype here, it starts with PH, and this is physical manifestation. It's kind of like a little mnemonic to help you. Genotype is all about the genes, and phenotype is about the physical manifestation. So those terms are going to be used constantly over many chapters as we're moving on throughout this. All right, that's the basics, terms that you need to know, but there's still a lot of process that we need to get to. We need to get from genotype to phenotype. Right, we need to get from gene to protein. And this is a really nice analogy that your book has about how this whole process works. And so pay attention as I step you through this analogy. The first thing that we're going to do is understand that we have these two terms, transcription and translation. And transcription is how DNA is copied. And that copy is going to be through this middleman called RNA. And then translation is how that RNA helps make the protein. So transcription, that's copying the DNA. Translation, that's making the protein. So your book has this great analogy. It talks about DNA as your grandma's cookbook. So you got your grandma's cookbook here, you got lots of pages in the grandma's cookbook. Maybe one is for making chocolate chip cookies, that's the one we're going to focus on. But maybe you got another one that talks about how to make, I don't know, banana pudding or something. And maybe you got another one here that talks about how to make fried chicken. I don't know, whatever your grandma makes. My grandma made fried chicken and banana pudding. Okay, so, but we're going to focus here on this gene. This is the gene, this is the page in the cookbook for chocolate chip cookies. And so here's what happens. You need to open the book. And in the process of DNA transcription, something has to open the book. And the thing that opens the book is something called RNA polymerase. Now, RNA polymerase does exactly what it says it's going to do. RNA polymerase makes RNA. RNA, poly means many, mer means units, ase means enzyme. So this green thing here is the enzyme that makes many units of RNA. And to do that, it's got to open up the DNA first. But here you see it making the RNA. Now, back to the analogy, your grandma, she loves you. She thinks you're the bomb. But she's not going to give you her cookbook. No, no, no. You have to make a copy of her recipe. So you're going to copy that down onto an index card that you can then, and your grandma, she loves you to pieces, but she is not going to let you cook in her kitchen. So you have to copy this recipe and take it home to your house. So the RNA polymerase opens up the book of DNA, makes the mRNA copy, sends that copy of RNA outside the nucleus. Again, all this purple is the nucleus, inside the nucleus, out into the cytoplasm. And out in the cytoplasm, we have the next big process that's called translation. And translation is, again, where we're going to make the proteins, or in the analogy, we're going to make the cookie. Now, there's lots of ingredients that go into making cookies. And so that's good. We're going to talk about all the ingredients that go into making proteins. including we need our mRNA, we need our ribosomes, we have these things called transfer RNA, we got these things called amino acids, all the ingredients. We'll become familiar with those ingredients. But eventually, if you do everything right and you follow that recipe perfectly, you're going to get some chocolate chip cookies. Or inside your cells, you're going to get your protein. So again, kind of keep that in mind for all the steps. I know it's a lot of terms I'm throwing at you right now, but this is a very term-heavy chapter. And it's probably the first chapter that we're learning real processes that are challenging. So stand by as we really dive into this process. And the first process that we're going to look at is transcription. Again, you've got some animations in your online materials. I'm just going to go through this with you step by step to talk about how DNA is copied. And we're going to make mRNA, or mRNA stands for messenger RNA. So there's 4 steps in transcription. 3 main steps, and then we've got some other really important stuff going on here too. So the first step is called recognize and bind, and what we've got is our RNA polymerase, this green object right here. And remember, we're still in the nucleus, so we've got this lovely purple background showing we're in the nucleus. And this RNA polymerase is going to bind to something called the promoter. I don't know if you can see that right there, but there it is, promoter. The RNA polymerase binds to the promoter. Guess what, guys? The promoter is part of the junk DNA. It's part of the non-coding DNA. What that means is that the RNA polymerase recognizes the promoter site, but it's not going to copy the promoter site. It's just going to say, oh, there you are. That's where, it's kind of like a, I don't know, like a bookmark. That's where I need to be. So that's all that recognize and bind does, is that the RNA polymerase binds to the promoter site of the DNA, and the promoter site is kind of upstream from the gene that we want to copy. The next step is the big step. This is called to transcribe, and transcribe simply means copy. It's like writing that recipe down on the index card. So we've got our RNA polymerase, and it's literally reading every base pair, at least on one side. Notice that it only reads one side. It's not reading this side, it's reading this side. And it is going to make a complementary copy. So for every adenine, we've got a thymine, and every guanine, we have a cytosine. And it just matches it up this way. I kind of just lied a little bit because I haven't told you something interesting about RNA, and that is that RNA doesn't actually have thymine. So before, when we were talking about DNA, when we had A's matching up with T's, adenines matching up with thymines, it doesn't quite work that way with RNA, because RNAs don't have thymine. Instead, they have something called uracil. The letter U stands for uracil. So we're going to match up A's with U's. So every time you've got an adenine, you're going to match it up to a uracil. And we can see the uracils in, I believe that's in pink. That's how you can recognize that it's different. And you see in the RNA, we have the pink ones, but there's no pink ones in the DNA. All right, so A's match with U's, and C's match with G's, and G's match with C's, and T's match with A's because mRNA totally has A's, just doesn't have T's. So a little weirdness there. We're going to deal with that during lecture. So that'll become hopefully really familiar for you. So again, we're still in step 2. We're just reading down this line and we're matching up the bases in that complementary way that we just talked about. Eventually we get to the terminate step. And the way that RNA polymerase knows to stop is it gets to this termination site, or it's also called a termination sequence. So it started at the promoter, it ends at the termination site, it just lets go. And when it lets go, it releases, kind of closes the book up a little bit, and it releases the mRNA, and the DNA kind of rewinds back up. It becomes back to the way it was before, at a double helix. But there is one more step, and it's called cap and process. And cap and process can be really straightforward, or it can be very complicated. So some of the straightforward things that happen to the mRNA is it gets a cap You can see here a cap and a tail. And so the cap and tail just act to help stabilize the mRNA. In general, mRNA is not a very stable molecule. It doesn't last for very long, maybe just a couple hours. And so the cap and tail help stabilize it. It also is going to help in the next step of translation. The other thing that has to happen are, if you remember those introns, these are the gray areas right here. Those gray areas are not coding, which means that it cannot stay in the mRNA. It has to go away. And so what happens is we call this like processing. We're going to remove the introns and string together all of the rest of that RNA. And we have this shorter molecule now, but this shorter molecule, that's completely coding. Everything there is going to contribute to the protein. All right, I'm going to stop it now, and when I come back, we'll pick up with translation.